Hi everyone. So far in this series, we've looked at the immune response and the action of antibodies. In this video, we'll find out how long-term protection is achieved. In the humoral response, we saw that cloned B cells can differentiate into antibody-secreting plasma cells and also into memory cells. These memory cells are responsible for long-term immunity in what we call the secondary immune response. When a new antigen is encountered for the first time, the specific antibody has not yet been produced. An immune response is initiated, as we've already seen. Phagocytes become antigen presenting to activate specific helper T cells, which in turn stimulate specific B cells to divide and differentiate, resulting in antibody secretion by plasma cells. All these stages take some time before the antibody can be detected in the blood plasma. This response following initial exposure to a new antigen is known as the primary immune response. Following destruction of the invading pathogen, antibodies will be broken down by the body, and so the blood plasma concentration of that antibody falls, as such high levels are no longer necessary. On any subsequent exposure to the same antigen, memory cells are already present within the circulation. These can quickly divide to give rise to plasma cells, which secrete the specific antibody in high concentrations. This is the secondary immune response. It's a much faster and greater response than the primary response. On the graph, we can see there's no delay before antibody concentration increases. The gradient of the curve is steeper and a higher maximum antibody concentration is reached. In our next video, we'll apply this knowledge of the secondary immune response to consider how vaccination is used to provide protection against infectious diseases.